Alright, welcome back. In this video, we're going to go over the loop of Henle or the Nephron loop. So, quick little thing. Thank you so much for almost 500 subscribers. We are 2 away. And yeah, really, thank you. I noticed that, I was looking at the statistics and like 78 subscribers came in from the last 28 days, which is amazing. I also noticed that about 70% of my viewers are female, so I see you. Okay, let's start. Before that, I also mentioned that for my big milestone, subscriber milestones, I want to make videos on drugs and whatnot. And I did say that one of the milestones, I will teach you guys how to make meth. I'm not joking. I'll teach you the synthesis of meth and actually how to make it for educational purposes only. But I'm gonna save that for like a bigger milestone. For the 500 subscriber one, I'll see. I'll think about something. What drug I'm gonna do, but it's gonna be entertaining. I promise. So let's get started. So in the last video of physiology, we went over the PCT, the proximal convoluted tubule, which was this stuff. So if you haven't watched that, go watch that. So now we're on to the loop of Henle. Now the loop of Henle has one primary function. It has one purpose and one purpose only. And that is to create a saline gradient in the kidneys, or specifically the nephron. Okay, so, and the reason we do this is because for the future parts of the nephron loop, this would be the DCT and the collecting duct. So in the next videos, we're going to go over the DCT and the collecting duct. And this is, what we're doing is basically setting up for that part. So that's basically the thing you need to get here. So the loop of Henle can be broken up into two separate parts. There's the descending loop and the ascending loop of Henle. So let's look at right here, this thing right here, this picture. So we have this portion that goes down, and then it curves back up and goes this way. So notice the arrows, right? The fluid goes down this way. It's coming from the PCT. It curves around and goes up, right? So the descending part's this, the ascending part's this, is that part. So we're going to break it down into basically two things, the descending and ascending, so it's basically easier to understand. Let's do the descending first. The descending is the easiest. It's by far, it is very easy. The descending loop, all they can do, all that can happen is water leaves. So we have that tube, right? So this came from the PCT. We have the tubular fluid. So inside this fluid, there's a bunch of stuff in there. We have a bunch of solutes, we have ions, we have garbage in there, we have urea, we have all this kind of stuff. We also have water in there. So in the descending loop, what's going to happen is water can leave. Water has the ability to leave through something called aquaporins. Okay? So what's going to happen here is first, I want to talk about the numbers in a second, but for right now, all you need to know is that we have the tubular fluid here, right? It's going down in this way, right? This direction. Water is able to leave the tube, right? It's able to leave the tube, and the water can join in the interstitial space, okay? Now, if there is excess water, some of it will be picked up by the vasa recta. It's basically like a blood vessel that neighbors the loop of Henle, and it's able to pick up water. Okay, so once again, just to reiterate, in the descending part, we have aquaporins. And aquaporins are specialized water channels. Okay, and what can happen is water is able to leave. Now let's talk about the numbers now. The numbers shown here is how concentrated that area is. So you notice that, okay, we start with 600 on the top of the tube, but then we go lower and lower. We increase in numbers, okay? So we go from 600 to 900 to 1200. So the numbers gain higher. Why? So remember, we have a bunch of junk in here. We have like sodium, chloride, we have potassium, we have urea, we have all these other junk stuff like steroids, a bunch of stuff, okay? Everything that your body needs to excrete out. Well, if this is a measure of concentration inside the tube, the more water that leaves, the stuff inside the tube is going to get more concentrated. There's less water in there. So if there's less water in there, that means the tubular fluid is going to get more concentrated. Does that make sense? So water is leaving through osmosis, okay? Because the outside space is more concentrated than the inside space. So by that definition, osmosis will take place. Water wants to move to locations of higher concentration. So this outside space is very highly concentrated of salt, of saline. So water is going to be able to leave, basically. And notice that the lower we get in, the lower we go down in this tube, higher concentration in the tube, because more and more water is leaving, meaning there's going to be more sodium, there's going to be more chloride, there's going to be more potassium, it's going to be more concentrated. So that's why the number goes up. Okay? Hope that makes sense. That's all you really need to know about the descending loop. It's just water can leave. Now the ascending loop is where it gets complicated. It's not too bad, but it's a little bit more complicated. So the ascending loop is where everything else can really leave except water. Water cannot leave in the ascending loop. So let's look at this picture right here. We're in the ascending loop, okay? So we're at this point now. We already did this part. That's the water. Oops, we already did this part. Now we're here, the ascending part. Our goal now is to actually make that saline gradient. Okay, so it's kind of weird. It's kind of weird how you think about it. You would think that, okay, if water is to leave, right, this area needs to be has a lot of salt, basically. But we're adding the salt in the ascending loop, not in the descending loop. So it's kind of weird, right? Water leaves first, and then we actually secrete the salt, the sodium chloride. So it's a little weird. I know it's a little complicated. But this in the ascending loop, everything else can leave except water. So our goal is to put sodium and chloride into the outside space. Not in the tubular fluid, but in the outside space. So how are we going to do that? Well, we're going to use pumps. Let's break it down. So this side is the tubular fluid. So this is the tube. So this is the stuff inside, this tubular fluid. The outside, or the destination, is the surrounding area, okay? And that's our destination. That's what we're trying to get at. So here's what we're going to do. Basically, we have cells. You have epithelial cells here, right? That line the tube, that make up the tube. They have channels in them. And so we're going to basically move some ions around. The first type of channel is called a symport. The sodium, chloride, potassium symport, all three. So sodium, 2 chlorides, that's very important, 2 chlorides, and 1 potassium from the tubular fluid is going to enter the epithelial space through the channel. Okay? At the same time, some potassium is going to leave and join the fluid. Okay, so not a big deal. Now, at the same time, we have the sodium and potassium pump going on. Now we have an influx of sodium. We have a lot of sodium inside, right? We need to shove it out. So we're going to use the sodium-potassium pump. The sodium-potassium pump is going to shove sodium outside the tubular fluid. So it's going to join this area here. Okay? Now we also have a lot of chloride inside, because I should have made that green, sorry. Let me put this as green so it doesn't confuse you. So we have a lot of sodium here. Now we also have a lot of chloride inside now, right? We have a lot of chloride. Okay, because we transported in with the symport. Chloride is going to naturally want to join sodium. They're best friends. I've told you this before in the last video. Sodium and chloride are best friends. Wherever sodium goes, chloride's going to follow. So chloride has its own pump. It's going to join sodium on the outside. So we're going to create sodium chloride. That's our salt. Okay? And potassium is also going to leave and join the outside, just cause. Okay? So we basically got we went over how sodium or sodium chloride or salt gets in the outside space here. And this, when sodium chloride or salt goes in this outside space, this can cause more water to leave, to get secreted out in the descending loop. So we can actually absorb more water. Okay? Now there's also the vasa recta that actually passes through this area as well and can pick up excess solids. So say if you have too much sodium and sodium chloride, we can't actually take this and put it back into the tubular fluid. We can't do that. Sorry, there's a motorcycle in the background, so I apologize. We can't actually if you have too much sodium chloride, salt, in this outside space, we can't just shove it back into the tubular fluid. We can't do that. But what we can do is put it into the vasa recta, a blood vessel, right? And it can be basically recirculated. So that's basically the loop of Henle. All you really need to know is that it's all basically controlled through a salt gradient, osmolarity and stuff. So know the numbers, know the relative numbers, and the more lower you go, the more concentrated it gets. And know the ascending loop and what basically causes, how do we actually get sodium chloride outside the cell. So that's basically it. If you found this video helpful, please like and subscribe. And until next time, later.